Welcome, I'm Professor Johnson, and in this video we are going to be talking about membranes and how we move things across the membrane. So first of all, kind of breaking it down for you, membrane transport, we're going to be looking at two main types. The first type being passive transport and the second type being active transport. Now passive transport can be broken down into more simplified versions. One is just simple diffusion and facilitated diffusion, and a special type of facilitated diffusion that we'll go into is osmosis. Active transport is going to require energy, and it's going to move in the opposite direction of passive transport. There's a special type of active transport that we'll go over known as coupled transport. And then the last thing we'll talk about is bulk transport, which is going to involve both endocytosis and exocytosis. So first of all, with simple diffusion is just like how a gas moves through a room. It's just going to be due to the collision of different particles that ultimately results in everything moving out. So you can see how those particles are dividing. If we took this to the extreme, we could imagine how all the particles would eventually spread out evenly within the place itself. Now, diffusion doesn't just stop, as you just saw right there. It's going to be constantly moving back and forth. There's just a few molecules in this that I'm showing, but it's going to be moving in both directions once it has reached equilibrium and the concentration is equal on both sides. So simple diffusion and all types of passive diffusion are going to move their concentration, are going to move particles down their concentration gradient. So they're going to go from high concentration to low concentration. Now, simple diffusion only works when the membranes are permeable to it. Cell membranes are semi-permeable. What does that mean? That means some things are going to be able to move across the membrane. The things that can move across the cell membrane are the small non-polar molecules. So gases such as oxygen, carbon dioxide, steroids, these are things that can diffuse across the membrane because they're small and they're non-polar. Things that are polar or larger molecules can't diffuse freely across the membrane, or at least not at a significant rate. And so they're going to need a little help, and that's what facilitated diffusion is. So facilitated diffusion uses protein channels and protein carriers to move things across the membrane. A channel is going to be something that just moves ions across. So it's an open pore that's selective towards the particular ion. So these channels can only recognize certain ions and will only let those precise ions through. Carrier protein is similar, except instead of ions, it's going to bind to and help carry specific molecules across the membrane. Now, you can have multiple ones of these across, and once it's through, you can have them diffusing through. You're still having the collisions like you always would. You can have more diffuse. Notice on that one, there was some moving back across the membrane. So remember, these things are constantly moving back and forth. It's just that their collisions are going to help drive them down their concentration gradient. So eventually you'll have the same rate of moving out of the cell as into the cell. And this is true for both just simple diffusion as well as facilitated diffusion. Now, there is a specific type of diffusion known as osmosis. So osmosis is the diffusion of water particles across semi-permeable membranes. Now, water can diffuse across the cell's membrane, but it does this very slowly. It actually is going to use aquaporins to help facilitate its movement across. So aquaporins are proteins that act as channels for water molecules. So osmosis is going to be the movement of water across these membranes. And water is going to be moving back and forth in both directions. So you can have diffusion across and forth. So osmosis isn't that interesting or conceptually until you start throwing solutes in there. So this solute here, that's what you dissolve into the solution, the solute right there. These solutes are going to help bind to the water molecules. This is why water dissolves these molecules, because the water's polar. These molecules typically are charged or polar themselves, and so it surrounds them and it helps dissolve them into the membrane. So this leaves you with two types of water. There's this water that's bound in there, and then there's this free water molecules. So these free water molecules can move back and forth. First of all, it's some terms. So the thing that has the more solute in it is known as the hypertonic solution, while the hypotonic solution is going to be the solution that doesn't have something in it. Now, if there's equal amounts of solute on both sides, which is not the case here, we call those two solutions isotonic. Now, the water is going to be diffusing through these membranes, and you're going to have it move in both directions. But the key here is all of these water molecules that are bound to the solute are not free to move across the membrane. And so you have what's the movement of water so that you have an equal concentration of free water on both sides of the membrane. And so it looks like there is more over water. And so you end up with more water overall on one side and less on the other side, thus the changing in levels. But it's an equal amount of free water molecules that's important here. So we can imagine having a cell that has a particular concentration of solute in it. If you would put it in an isotonic solution, nothing would happen. You would have the similar concentrations on both sides. However, if you put it in a hypertonic solution, the water is going to diffuse across the membrane. The ions won't be able to if you don't have the particular channels for it. So if you put it in a hypertonic solution, water is going to move out of the cell towards where the more concentrated portion of the solid is. So you're going to have it moving out and it would end up shrivel. If you put it into a hypotonic solution, on the other hand, because there's more solute in the cell, water would start moving into the cell. And there are certain situations where it can move into the point where it would actually burst. So that was it for the passive type of transport. Let's look now at active transport. So active transport is different than passive transport in two main ways. The first way is it uses ATP. So that's what these three red balls here represent. They represent the three phosphate groups on an ATP. So it's going to require energy. Why does it require energy? Well, unlike passive transport, active transport is going to be actually moving things from a lower concentration to a higher concentration. So it's going to move it across the concentration gradient. So how this would work, so you would have your molecule that you're moving across the membrane. It's going to bind to the transporter that is going to require energy. So you have hydrolysis of ATP. You're breaking this into ADP and an inorganic phosphate. And when it does this, it changes the protein's conformation such that you can move it out of the cell. And you can do this sequentially several times on the same protein and eventually move the concentration to a very high concentration on one side and a low concentration on the other. Now, there is a specific type of active transport known as coupled transport. Coupled transport is going to use ATP indirectly, and let's look at how this works. So you have this transporter over here that's your coupled transporter. It binds to something that's going to be moving down its concentration gradient. Notice that we had all these stars here that were moving up their concentration gradient. In the previous one, in this image, they're moving down their concentration gradient. Now you might have, or you would have, this other molecule, which is represented here in red. You can see that there's more of it over here than there is out here. So it is actually moving against its concentration gradient. So it's using the potential energy of something moving down its concentration gradient to help move something up its concentration gradient. So once you have all three of these things bind, you would have a conformation change in the protein such that everything gets moved in. Now, this is coupled to your traditional ATP or traditional active transport proteins, in that you would have this transporter here that would be moving it against its active transport using the ATP with it. So that would be how coupled transport works. Finally, with both of the facilitated transport of passive diffusion, or facilitated diffusion of passive transport, and with the active transport of all active transport, where you have proteins in the membrane, one thing that can happen with these is that these proteins can become saturated. That is, they can bind so many of their molecules, they're moving across the membrane, that they can't keep up with them, and there's more molecules to bind. So you can imagine a situation where there's not very many molecules that need to move across the membrane, and then they might have plenty of time to move across the membrane. So they have plenty of time before it reaches there, and so you have movement across the membranes slowly. Now, you can also have a situation where there's a lot of proteins, and here you could have things moving through, but you can see they're trying to move through there too rapidly. The transporters here are saturated in that they can't get everything through there at the same time, and so that's something that's characteristic of transport that utilizes proteins, is that it can become saturated. Whereas transport that does not use proteins won't become saturated, because it's just diffusion through the membrane. All right, the last thing that we're going to talk about here is bulk transport. So there's two main types of bulk transport. The first one is endocytosis, which is the bulk intake of substances into the cell, and the second is exocytosis, which is the bulk movement of things outside of the cell. Endocytosis can be divided up into two other kinds. One is known as phagocytosis. So phagocytosis is what is going to happen when you have actual solid particles that are going to move through the membrane. The other type is penocytosis. So penocytosis is referring to drinking. So this is when what you're taking in the cell is liquid, either a solution or water itself being moved in through the membrane, taking it in. Now, endocytosis can be specific. You can have these different proteins on the surface of the cell which are going to recognize specific solutes that you're going to bring into the cell. So in that way, it can be specific to the things that you're actually taking into the cell. The other type is exocytosis. Here we can see at the end of a synaptic terminal, this is where your signaling of your nerves is occurring. You can have these vesicles that contain these different signaling molecules, or it can contain other things as well, and they can move to the surface of the cell and just be released in bulk. So this is exocytosis. It's just the bulk sending out of particles from the cell. So that's it for this video. I hope you learned a lot about membrane transport, and I will be seeing you guys in class.